To have a really outstanding panel, and to introduce that panel is Mason Freeman, who has experience really on both sides, on the industry side, on the academic side, and who's currently the director of the Translational Research Center at Mass General Hospital and a professor of medicine at HMS. Mason, welcome to you and to the panel. Thank you.
Good morning, everyone. Thank you for coming to the early bird session. You will be rewarded for that early rising. Our topic is innovation in clinical trials, and I was here on Monday hearing about the heart failure trials, and many of them have not been the outcome successes we had hoped. And we've assembled a panel here today that actually has had real success in doing major cardiovascular outcome studies, and I think that the conversation you're going to hear from them today will give you some insights into what the success stories have been and also some of the challenges. So let me start by introducing the panel. To my left, Seigo Izumo is the Senior Vice President and Global Head of Regenerative Medicine at Takeda. Jessica Mega is the Chief Medical Officer at Verily, better known maybe to some of you as the Life Sciences Division, formerly of Google X and Alphabet. Alan Moses is the Global Chief Medical Officer at Novo Nordisk. And then at the end of the row is Pretorius, who's the Chief Scientific Officer at PAREXEL. You can see we're a rather eclectic group here. I think that probably harkens back to the same adage in the military, which is that war is too important to be left to generals, and cardiovascular outcome studies are too important to be left only to cardiologists. We have an experience here with some of the clinical trials in cardiovascular disease that focus primarily on metabolic products, that though they require cardiovascular safety outcome, we'll be talking about that. And the challenge that we're facing with those, as well as other cardiovascular trials, is twofold. One is a great thing in medicine today, which is that the standard of care, particularly atherosclerotic cardiovascular disease, has improved dramatically, and certainly in my lifetime, with the advent of lipid-lowering therapies, better hypertensive therapies. So the event rates in cardiovascular trials have been declining very significantly over the last few decades. And if you're probably, as you've just heard a little bit from Rob and Joe, if you're planning on a cardiovascular outcome trial, you deal with the reality that the event rate often is in the 3 to 4% range for the major cardiovascular events that the FDA cares about in these trials, which necessitates a very large trial, often done for a very long period of time. So this is one of the great challenges and the great expenses of cardiovascular trials. Most of them now probably cost anywhere between $500 million and $1 billion. So making an investment in that, as I think you just heard Rob say, for the bigger pharmaceutical companies is certainly a major decision. And in a world where I spend some of my time, which is starting new companies in the venture capital world, we almost never make an investment in a metabolic company now for diabetes, simply because the cost of the cardiovascular safety study that's required is not something that a venture capital group with its limited resources can actually pay for. So this becomes a huge barrier as we get heavier and we have more diabetics. We are actually choking off the early stage pipeline because of the cardiovascular safety requirement in a way that I think we really need to take cognizance of in our regulatory environment in the world. So the panel today is going to be talking about a variety of these different issues, ranging from how we handle the increasingly large data sets that come from these enormous trials and the new information that's coming from different kinds of devices that can be used to capture even more data. We'll be talking a little bit about trial design and ways that perhaps trial design can improve the ability to do faster, more efficient clinical trials. And we're going to be talking quite a bit about technology. And then we have Seigo on my left, who's an old friend and dear colleague, who's actually going to say, I don't really want to do very large cardiovascular trials. I'm going to be focusing on something in a smaller subset of people with perhaps a very innovative therapy. So he will bring an entirely different perspective. So with that, I'd like to start with Alan. His company has had the great fortune of developing one of the really impressive diabetes medicines, and in its cardiovascular safety study actually found a cardiovascular benefit, which was really a remarkable achievement. And with Alan, I think you had some insights into how that was achieved and what it took to be able to do that study.
Well, it's actually interesting, Mason, because in 2016, we completed 3 cardiovascular outcome trials in the diabetes space, 2 on GLP-1 receptor agonists and 1 on insulin that represents the first head-to-head double-blind study of 2 different insulins done as a pre-approval requirement in the US, although not in Europe, to get insulin degludec or Tresiba approved. And each one represented a different sort of challenge because from a regulatory perspective, which has driven so many, really almost all of the CVOTs we've seen in the diabetes space over the last 7 or 8 years, one of these trials was a post-marketing commitment to assess both safety, cardiovascular safety, and general safety. Another was a planned pre-approval assessment for a new drug that's currently under regulatory review. And the third trial, the insulin trial, was also a pre-approval study with an interim analysis that required a whole different setup for us as a company and for me personally, but that's a separate story, in terms of being able to assess the data and not compromise the continuation of the trial. So each of these was slightly different in size, but through each of them, we have learned a great deal about conducting CVOTs. And one of the most important things, of course, is making sure you have the right sites. And even more important than that is making sure that the subjects are retained during the course of the trial so that you have integrity of the data set with very little missing data over time. And through active collaboration with academic investigators in the steering committees, we've been able to accomplish that in a way that we would never have envisioned before starting these trials. And each one has been more successful than the last in terms of completers, minimal loss to follow-up information. We're talking about in our last trial, less than 10 subjects lost to follow-up or with unknown vital status at the end of the trial. Why is that important? Because from a regulatory perspective, the FDA and EMA are both increasingly concerned about missing data and the statistical approaches that one needs to use in order to compensate for missing data that may occur. So engaging investigators, engaging patients in approaches that use standard of care methodologies that are appropriate, obviously, for the given geographic region, and that's another challenge for these global trials, where standards of care do vary a little bit across countries, across continents, is increasingly important. And we've found that by engaging investigators, national leaders for each of these trials, in a global expert panel, and then having local expert panels and local panels of nurse coordinators has made a huge difference in terms of being able to provide information to investigators once they've started in the trial, provide mechanisms to engage patients throughout the course of the trial so they stick with it, even if they drop drug. And in the end, result in data sets that are much easier to evaluate in regard to the absence of missing data to make the data interpretation a lot more clear and the conclusions more obvious.
So let me just quickly ask a question there about technology. Are these requiring new technologies for interacting with the patients to retain them? And is this something that Jessica and Verily is going to be helping companies like yours to be able to do more of in the future?
You know, it's interesting because I do think there's a major role for that in the future. But for the cardiovascular outcomes trials, we've been less inclined to use the technology that we're starting to use in the diabetes metabolic trials. just because of the nature of the signal. But engaging patients throughout the process, I think, also can benefit from the use of electronic tools, which we have not done to date, but maybe an opportunity in the future.
Jessica, I think that's an opportunity to transition to you about how you're using data and technology to pursue your interest in cardiovascular and other health conditions.
So it was very interesting. One, it's fun to be back. I spent most of my career at Brigham and Women's Hospital and at Massachusetts General Hospital. And what's been very interesting in the field of cardiology is the fact that we are privileged that we have such large clinical trials. But it became quite clear to me when I went to visit some of the traditional tech companies that there are tools out there that we could be deploying in these trials. And in fact, I think it's a necessity. Rob mentioned in the last session the idea of trying to figure out ways to streamline cardiovascular trials, and I think you brought it up this morning. How do we make sure we don't squeeze the pipeline? And I see two major trends. One is, how do you engage those patients not only in the four walls of a clinic, so someone comes on site and we see them, but how do you provide tools where patients are really the center of the trial? So patient-reported outcomes become center. Sensors may be very important depending on what your endpoints are. And in fact, we may even change the concept of the endpoints. There's always going to be hard cardiovascular clinical outcomes that are important, but we also talked this morning about what does it mean to improve someone's functioning. So I think this idea of really understanding people in the 364 days of the year that they're not seeing us in clinic for a trial is really important. The second thing is really coming up with back-end architecture and infrastructure where information can be coming in real time. We can analyze it, and I'll just give a quick anecdote is the idea of trying to be I was part of the TIMI study group in M, and it's the concept of how do you get data in and not have to turn it into a PDF and circle different values and then remember them. Why are we not demanding that we want real-time dashboards that help us analyze the data so that we can have adaptive trials? And that's one thing at Verily we've been spending a lot of time on is how do you create the tools to help understand people in the world, and how do you create this back-end infrastructure that can deal with real-time sensor data, molecular data, clinical data, claims data, and make it accessible so that we can get back to the science. So I think that there's going to be a real opportunity to accelerate cardiovascular innovation.
I would suspect that the technology you're describing are generating huge new data sets in addition to the ones we're used to generating in clinical trials, which are laboratory values along with a variety of other parameters that we measure in those trials. Is your company working on presentation of these large data sets that enable the poor investigator sitting in front of a 10,000 patient trial looking at liver function tests and trying to figure out whether or not there's a signal inside of all that noise? And how do you envision being able to aggregate all that data in a platform that can be useful for the clinical investigator struggling with these huge sets?
Yeah, so two things I'm saying. One is, how do you deal with multidimensional data? So it's one thing to have the liver function test. How do you merge that seamlessly with the clinical outcomes? How do you deal with digital readouts? And so there's a huge effort in doing that. So that's one piece. And then secondarily, this idea of what we even define disease like today may be quite different. And so in the we're not at a neurology conference, but people are starting to say, why would you measure a tremor, for example, when you see someone once a year or a few times during the year for a clinical trial? Why wouldn't you instrument everyday devices, something like a spoon, and measure tremor every day of the year? Mm-hmm. And so I see big changes in that regard.
Fantastic. Sai, we're going to turn to you to solve one of our great challenges. Most of these cardiovascular outcome studies have a fairly traditional clinical trial design. A linear start till there's a certain number of events or until a certain duration of therapy has been given. There's always a lot of talk when we talk about innovation in clinical trial design about new ways of doing it, adaptive trial designs, other designs. As somebody who's leading one of the largest CROs in the world effort in this area, do you see those new trial designs, and which ones do you find captivating as potential solutions to some of these difficulties?
Absolutely, Mason. I think first and foremost you mentioned adaptive trials, which is something that I'm very passionate about and that I passionately believe holds great promise for the future. So maybe just to frame it a little, what is an adaptive trial? And there is a simple analogy that I like to use which explains it, and it compares sort of the traditional trials that you outlined traditional randomized controlled trials as cargo planes without any windows. Analogy being, you of course cannot have a look at the data during the trial, while the plane is in flight. We basically have to wait until the plane lands, the database is locked, and we get a sense of what really happened during the clinical trial. Adaptive trials, in this analogy, are more akin to passenger planes. So you have these windows, these opportunities during the conduct of the trial to take a look at the data and to make some prospectively planned, and that's the important part, some prospectively planned course adjustments, which is incredibly valuable. And I believe these trials hold a number of benefits, to highlight maybe one or two. First and foremost, in my opinion, the big benefit is the flexibility that it offers. A lot of clinical trials fail. If we look at the phase 3 trials, the number being thrown around is around about 50% of these fail. And before we start these phase 3 trials, when we plan them, we make a ton of assumptions. And having an opportunity during the course of the trial to revisit those assumptions and to make some course adjustments is incredibly valuable, and I think has the potential to really change the outcomes of many of these phase 3 trials. I also do believe these adaptive designs offer more value per dollar invested. What I mean by that is there is a common misconception that adaptive designs are cheaper and faster, which they are not. But I do believe over the entire drug development life cycle of an asset, they do add value and they do help to alleviate some of the cost and time challenges. And then the final benefit, I would say, is, in my opinion, they are more ethical. Recent example, we were running a trial in which the selected dose was clearly inefficient. And so exposing patients to an extended period of time to a dose that's not efficient is just not ethical. Having an opportunity to adapt would have been very helpful in that scenario. Maybe to wrap up, you mentioned some of the other interesting designs. We are certainly seeing a lot of that. I do believe that the whole omics revolution and the rise of precision medicine have yielded a number of very innovative targets, a number of very innovative therapeutic modalities, and also a number of innovative clinical trial designs. To name a few, we see these umbrella designs starting to emerge quite a bit. Umbrella design being where we have a number of different treatments against a single indication in one clinical trial. We're also starting to see basket trials, which is sort of one treatment against a basket of indications in one clinical trial. And then even some of these newer platform designs, which are a number of treatments against a number of indications in one clinical trial. And I think the main benefit of these is, relatively speaking, clinical trials from an infrastructure perspective are inefficient, meaning we build a whole infrastructure, sites, investigators, patients, a whole host of regulatory documents that goes with it, and we only use it once, typically in a standard design. And some of these more innovative designs give us an opportunity to use that more efficiently in evaluating a number of drugs in a number of indications.
Can I ask you about the interactions that you have with the FDA and the regulatory agencies when you move to these newer designs? Oftentimes there have been concerns about statistical power loss, perhaps some potential for unblinding in the study with these approaches. What has been the reception, and maybe others on the panel can comment with their own studies about that, but could you comment about that, Sai?
Yeah, I think you touch on a critical point, Mason, and that is you have to work with the regulatory agencies, with the FDA, with the EMA, when you're considering these during your development cycle. And even though I am passionate and a big proponent of these more flexible designs, they have to be used appropriately, meaning an adaptive design is not an excuse for a poorly designed trial. So you have to use them appropriately, and there are there are diseases where these are probably more appropriate. Typically, we are looking for a disease or an outcome that has a pretty rapid readout. There's no sense in adapting if you have to wait 5 years for an overall survival rate. So you need something that has a pretty rapid readout. So need to interact with the regulatory agencies. And I have to say, contrary to what I'm hearing in industry, the regulatory agencies, certainly the interactions we've had, have been very supportive of these and have come out publicly. You know, Janet Woodcock and others, I think Rob just mentioned it in the previous session as well, supporting these types of innovative designs when used appropriately. Final comment maybe in that regard, Mason. What we're seeing at the moment, both based on our own data, but also based on data from Tufts and some of the other sources that are looking at this, is that there's a lot of, this is being adopted quite heavily in the earlier phases, phase 1. In the phase 2-3 space, adoption is about 20 to 30% of all trials currently running. So it is happening, and I think the reluctance, as you quite rightly pointed out, is when you run your confirmatory or phase 3 pivotal trials, people are hesitant sort of doing something out of the box when it comes to that. But I certainly think if you plan it appropriately, if you simulate your type 1 errors, what you alluded to, make sure you maintain the blind, only people that are allowed to see what they should see. There are ways to ensure data integrity throughout, and if you can reassure the regulators about that, these are very appropriately and well received.
Great. Alan, did you want to comment about that?
I was just going to comment about the interim analysis, because I think that's a uniquely challenging situation from trial conduct and regulatory evaluation perspective. I had mentioned one trial that is actually going to be presented at the ADA in about a month from us, which is a double-blind comparison of 2 insulins that was required by the FDA as a pre-approval commitment based on a signal that they believed, although we did not, represented a concern for a particular insulin degludec. And so we had agreed with them, and this was an intensive negotiation, that we would conduct an interim analysis, and that's fine. What I think were naive about at the time that agreement was made about what exactly we would have to do to conduct and then protect that interim analysis. And that turned out to be rather challenging. I think it was an extraordinary experience for us, and we have benefited from it in the long run, but it points out the challenges of having data and of the necessity of keeping those data incredibly close to the vest until the trial is completed, so you don't compromise the conduct of the trial until completion. And fortunately, the outcome was very good. The drug was approved based on the interim analysis, but we needed to make sure that the trial could be completed without that compromise.
All right, now we're going to come back and talk about some of these clinical trial issues in more depth, but this is your opportunity now to think about questions to a panel here that has an enormous amount of experience in conducting these kind of trials. We're going to open this up in a few minutes to questions from the audiences, but at the moment we're going to shift to Seigo, whose group is working on some very exciting technologies for stem cell and regenerative medicine in the heart, which may be one of the solutions to the heart failure problems that we were hearing about before. And Seigo, why don't you tell us about this exciting work that you're doing?
Yes. So at Takeda, we made a major commitment for the clinical application of iPS cell-induced pluripotent stem cells. Some of you know that these cells are discovered or invented by Shinya Yamanaka of Kyoto University, who get a Nobel Prize for this discovery. Basically, blood cell or skin cell can be taken from patients and or a normal volunteer, and they can be converted to any cell type, including beating cardiomyocytes, beta cell, just to name a few examples. And at Takeda, some of the projects we are working on is development of a cell-based therapy for the severe heart failure. So the paradigm is in this case, it's allogenic. Dr. Yamanaka and the Kyoto University is building a master cell bank that has, derived from HLA homozygous donors, so that we have a majority of the HLA haplotype covered that are commonly seen in Japan. And you make cardiomyocytes, not only just cardiomyocytes. If you make cardiomyocytes from iPS cell, you have a mixture of atrial cell, nodal cell, ventricular cell. The Dr. Yamanaka and his group has a proprietary method to purify ventricular myocytes so that we can get a more electrically homogeneous population. And we envision that patients with severe heart failure otherwise would have qualified for cardiac transplant will receive the cell therapy directly injected in their heart. In Japan, for a variety of reasons, cardiac transplants are done in a very small number. The latest number is about 30 plus, maybe less than that MGH and Brigham combined do every year. And also, LVAD is implanted far less frequently. So there is a need that are alternative therapy for the severe heart failure. And obviously, in this setting, that you cannot do what is very difficult to do, traditional randomized control trial. So Japanese government and the regulator, PMDA, it's a Japanese FDA, has opened a new path, the Regenerative Medicine Act, that allow the development of a cell therapy, gene therapy product. And after phase 2, if you demonstrate a significant beneficial effect on surrogate endpoint, not the clinical outcome endpoint, in the case of heart failure, that would be ejection fraction, functional status, patient symptom, for example. And not necessarily have a control or placebo control because the open chest procedure would be difficult. So this is more akin to development of a surgical procedure. As we all know, that major cardiac procedures came into practice without going through a traditional randomized trial. And despite that, we know that the procedure can be improved and the procedure can be proven to be effective. So the first case of such a conditional, and then the regulator require all the patients got into registry and follow up. So the first case of such approval actually was done recently. In this case, actually, autologous skeletal myoblast. The cardiac, the sheet was made and transplanted to patients. 7 patients underwent such procedure, and all had improvement in ejection fraction, functional status after 6 months, and the Japanese regulatory agent gave them a conditional approval. The company have to do, this is actually, by the way, it's a thermal company, not Takeda, have to enroll, additional, 100 patients over the next 5 years, and have to show true outcome on the mortality improvement for the product to stay in the market. But anyway, for severe patients and modality like a cell therapy, perhaps that are alternative way, more like akin to surgical procedure development, may be the way to bring the product to appropriate patient population.
And I presume that these studies have to be done at some pretty sophisticated centers in Japan with these patients. Is that the plan right now, to conduct them at tertiary academic centers in Japan?
Yeah, right now that the first case skeletal myoblast trial was done only at the Osaka University, the largest transplant center. But we envision that when we are ready for iPS cell transplantation, we would use probably 2 or at most 3 centers to start with.
Great. And you can imagine this technology being able to scale to much larger numbers if the efficacy is there?
If efficacy is there, right now that we are thinking of at least under direct visualization, some kind of a thoracotomy, or maybe using a scope. But if safety and efficacy is demonstrated and sufficient, there's the continuous improvement in technology to grow these cells in a very large number at a lower cost. One can envision that perhaps using a catheter like a Noga catheter, you can inject the cell by the catheter. Then I think there's a chance to be applied to more patients, not restricted to a few cardiac surgery centers.
Great. Terrific. It's very exciting, Seigo. Let me turn. We're starting to get some questions. I'm going to ask the panel a question that came up this past week that I think caused quite a bit of consternation in the clinical trial world. There was a letter from the investigators from TopCat. I'm not sure if it was discussed at any point earlier in the meeting, but there was a TopCat, as you may recall, was the use of spironolactone in heart failure patients with preserved ejection fraction. And the letter came out reporting that in Georgia and Russia, 30% of the patients had no drug metabolite in their bloodstream. The investigators did not comment as to how they thought that might have transpired, but it certainly raised the issue about quality control in clinical trials. As we do these huge clinical trials, the latest PCSK9 studies that were done internationally had 27,000 patients in them. So finding that number of people to be involved in a clinical trial is difficult in and of itself, and given the cost of recruitment, we have naturally tried to migrate to some places in the world where the cost might be a little less than the US. And I wonder if others on the panel have comments about the viability of those approaches, and is there any way for us to be able to monitor data to pick up early signals of behavior in trials that we might like to avoid? Anyone want to take that challenge on? Sai?
Yeah, I think it's of course a very topical issue, and one that for the last few years have been discussed quite extensively. There is an approach, and I think it's one that has been implemented in the clinical trial process for many years in a variety of ways, called data surveillance, or at least that's how we refer to it. And it, in essence, enables you to look at the data in a blinded fashion on an ongoing basis. Not just operational data, so cycle times, our sites sort of keeping up with their source data, etc., but also AE data in particular. Are there countries where we don't see any AEs, for example? And we use a very sophisticated advanced analytics system to monitor these things on a real time and set margins around it. Because what you don't want to have happen is exactly what you just said, come to the end of a trial and say, well, there weren't any adverse event in these two countries, which doesn't make sense, or why are these body weights at the site all pretty much the same, et cetera, et cetera. So all examples of red flags that could have been and probably should have been picked up earlier in the process. So data surveillance, I do believe, is a must for the future, and something that certainly we've invested a lot in. And some of the analytics that Jess mentioned comes in very handy there.
We've had the good fortune of dealing in a therapeutic area, diabetes and cardiovascular disease, where we have a biomarker that provides evidence of people taking the drug or not taking the drug because it has a glucose effect. And that makes it a little bit easier. But I also want to comment about the geographic distribution of these global trials, because we are a very global company, and all of our trials are conducted globally. It turns out that in most cases, adherence to protocol, adherence to medication is actually better outside of the US than it is in the US. And that's a real challenge. In part because, of course, the FDA in particular likes to see a reasonable percentage of individuals from the US. Two, we believe we're dealing with sophisticated investigators and sites in the US, and yet when you actually look at adherence levels to drugs, it's actually probably twice as bad, if you will, as it is in other parts of the world. So it's a constant battle to find the right sites and the right motivation for both investigators and subjects to continue in the trial. And I think this, maybe Jesse, you can make a comment about this, is where some of the monitoring devices or approaches may be more useful over time in particular.
Yeah, Jess?
So I think it's a critical issue if we're putting all of these resources into a study. And in fact, if we're enrolling patients, we have an ethical obligation to make sure they're run with the highest standards. And so I'll give two tangible examples. One is in terms of what infrastructure is needed to be able to do real-time monitoring. We're engaged with a partnership with Duke and Stanford to run what's called the baseline study, which is a longitudinal epidemiologic study. And on one hand, we're quite interested in this very deep data repository, and I'm happy to chat about that. But the other piece that doing this kind of study, the other piece that's so important is really understanding how an individual in a study interacts with the data from start to finish. So people come into this study, they log in through their own account, so they are put in the center. So it'd be nearly impossible if you use these types of tools and if the patients understood what was going on, that they wouldn't be, for example, taking their medicines. So people are coming in through their own portal, the consent is there, they then are going to be engaging with different devices, and so we could get to a point where we think it's so important that maybe patients need to take a picture of their pill every day. We would have to decide what's relevant. But all of a sudden, when you have that two-way communication with individuals, things don't happen in a black box. On the back end, if you can then watch the data flow in real time, you can do the exact monitoring that I was talking about. And in fact, monitoring takes up a huge amount of resources in current clinical trial design if you're doing 100% source document verification. If you can start to look for patterns, the example of everyone at a site has the same weight, everyone has the same electrocardiogram, all of a sudden fraud can get detected much earlier. And if you look at a lot of these large informatics companies that are looking for fraud detection broadly, they're looking in many different spaces, but the same can be done here. But I think it can be done in a really positive way. It's not Big Brother's looking after you, it's looking for data quality. And so we are quite interested in coming up with architecture that people can use broadly so that we can do these trials and do them in the most ethical way.
Is there a regulatory control in terms of good clinical practice around the use of this kind of data that is evolving? We have that kind of control over most other kind of data sets we have. Is the new technology that's being utilized to track and follow patients that might be ending up a parameter of clinical outcome or benefit of the drug? What are the regulatory controls that we are dealing with? Is that established, not established? Where are we in this, Jess?
Well, one thing that's been incredibly fun is most regulatory agencies are quite interested in how do you use the next generation of tools. I've actually found people to be incredibly receptive to try to say, how can you come up with compliant? One thing that's very important, for example, is that you know the data comes from that individual. And so what kind of techniques can you use to make sure that it's not, that I don't take this device and put it on my child and determine that I actually got a lot of steps that day. But there are a lot of interesting fingerprints that you can try to use to make sure that these are all done in the most compliant way. And I think there's just a lot of energy you can see from people in the room trying to figure out how do we make these trials as efficient as possible.
Yeah, Sai?
Maybe just further to what Jess said, a practical example, we're running a clinical trial now aimed at validating some of these wearables, and it's in public domain. We issued a press release. The wearables being, sort of a pulse oximeter, ambulatory blood pressure, glucometer, activity monitoring, spirometry, and also body weight. And as Jess pointed out, the challenge is not just capturing and validating the capture part of it, but that whole data flow, data analytics, data visualization piece, because these are massive data sets, as you quite rightly pointed out. And validating that, to your question, Mason, in a way that is both HIPAA compliant as well as 21 CFR Part 11 compliant.
And there's another element there, and I wonder if Sai and Jessica could comment about this, and that is the issue of real-time monitoring acting to unblind a subject as to which arm they're in. And for certain of these trials, the regulatory trials in particular, that is a huge risk.
I'm happy to chime in. I think you're right. We have to be very careful that the readout that we're seeing is something that is shared with the right group in the right context so that we don't unblind. And I'm a huge fan of, really, randomization is so important because we think that we know all of these observations, and then you randomize. And I think in the cardiovascular world, we've been burned a bit by relying on surrogate outcomes and thinking that we understand how things are going to unfold. But the other piece that's going to start to happen, and we're seeing this in research broadly, is participants are going to want their data back. And they are going to be the stewards of their own data. And how do we share data, put the patients in the center, but then also keep these trials blinded? And so I think we're going to have to start to get prepared, not only creating the tools where you responsibly give the data back, but making sure that we can run sometimes trials that take several years in a blinded fashion.
Seigo, you have a comment?
So these wearable devices are not only useful during the trial, but also very useful for the post-approval registry. For instance, registry. In the registry that we call patient back, every month or every 3 months, and how you are doing, and you write, in New York Heart Association 2-3. But we're going to have, particularly the small number of patients, let's say severe heart failure patient underwent cell therapy, we can monitor them all the time. And I believe that a much better quality of the data on the patient functional status can be obtained that way.
Okay. I'm going to give the audience a chance to get their questions responded to at this point. So I asked this question with some hesitation when I was on a sabbatical at Novartis. Everyone who was in charge of biomarkers lasted about 6 months and got fired because they never had one that actually worked. And when they asked me to be the global head of biomarkers, I realized they wanted to get me out of the company. So that was my experience. But we have a question from Anthony in the audience who says, do we see any biomarkers in these areas of cardiovascular outcome studies that look promising that are not yet in our routine use at this point in time? Alan, anything in the trials that you've done at Novo that has gotten you excited about something that could actually make a trial more informative earlier with fewer patients because you've measured something that you thought was relevant?
I wish I could say yes. We've looked very carefully, and we've not done a great deal in terms of atypical biomarkers in regard to the cardiovascular outcome trials that we've conducted. But right now, the best predictor of response is what the response is, unfortunately. And part of that, I think, depends on the fact that as we're trying to figure out the mechanisms by which new classes of drugs, particularly the GLP-1 receptor agonists, act, it's very difficult to rely on a biomarker when the pathways that are affected, and there are multiple pathways that are affected, actually directly relate to the cardiovascular outcome. So we're very interested in looking, and I think one of the areas that we're becoming much more involved in, particularly non-alcoholic steatohepatitis or NASH, is going to be ripe for the development of biomarkers that can be insightful. But so far in the cardiovascular outcomes, we haven't found them.
Okay. Sai, can you save us here?
Well, I think completely agree with Alan. Nothing that just jumps out in the cardiovascular space. Although I would say when I think about biomarkers, especially in the context of the topic today, translational research, they do play an incredibly important role, an increasingly important role in sort of making sure that we can show that that target is being modulated, that there are downstream processes. So not as an outcome, but sort of checking one of the main pillars in the early translational space. What I have seen, what I think is also relevant to the question and important is on the regulatory space. In the regulatory environment, we've seen a significant change in terms of being more receptive and accepting of these as outcomes. Case in point, about 3-4 weeks ago at the end of March, the EMA, the European Medicines Agency, issued a draft guidance for the use of fibrinogen as a surrogate marker, an outcomes marker in COPD trials. So I think it's changing rapidly and it's moving in that space. Hope and optimistic that we'll see similar progress in the cardiovascular space.
And Jess, how about from the data analytics side? Is there something that you think is emerging, or others, or that you?
I think so. It's very when I traditionally thought about biomarkers, I thought about blood-based biomarkers, and that I think is still a very fertile field. This idea that we lump people based on ICD-9 or 10 codes just still seems rudimentary. But I want to plant the seed. I have now started to think about biosignals much more broadly. And so the idea, at the Precision World Medicine Conference, almost all of the talks were based in genomics, and I think there's still a huge amount to do there. I personally spent time looking at the connection between genetics and drug response. But now that we have digital readouts, and we'll have to see what that means in cardiology, but I mentioned in neurology, for example, if you're looking at movement disorders, all of a sudden, all of these readouts, I think, are going to change the way we classify disease and the way we understand disease. And in fact, the trials are starting to get smaller because, again, you're not measuring. I gave the tremor example. On the cardiovascular side, potentially, if we're looking at blood pressure medications, medications that affect QT prolongation, these types of things, if we think about biosignals broadly, I do think that we're going to start to see some movement.
Great. Jess, I'm going to come back to you from a question from Samir. When I first read this, I thought he just wanted you to change his password because he can't log into his email account. But I've read it 3 times now, and I think that's not what he's asking. What he's saying is, I wonder why your company, Verily, doesn't get into the electronic healthcare record business and save us, and this is a pun I'm sure intended, from our misery of epic proportions. Is there... is there any ambition or goal in Silicon Valley to help us out and be able to use a medical record that's a little bit more intuitive and friendly to, as I think I heard Callum say, we data entry clerks now that used to be physicians?
So 2 things. If you need help with your password, please find me afterwards. Secondarily, one thing that I've enjoyed, this concept of user-centric design, and we've started more on the patient side. What does it mean to create tools that people actually want to use? And going back to the baseline example, it was fascinating. I spent as much time working with UX designers as I did with clinical researchers, because if you create something that no one wants to use, they're not going to log in, and they get stuck. We have this thing called Bugganizer, and so people will test the different products, and people will submit all these bugs to make sure you can seamlessly go through an experience. The same is true for the devices that we're creating. I think physicians, as I'm reminded at work, physicians are people too. So we probably need to start to think about user-centric design there, but I would say we're starting really with patient-facing front-end tools and then data infrastructure on the back end.
So if I could just make a comment about that, because I've been privileged over the last 4 years to live in Denmark. Having moved from Harvard to Denmark, it's a strange sort of experience. But one thing that I have experienced, both in working with the Scandinavian countries and also specifically with Israel, is that the integrity of the data that they have at the patient level across populations just so far surpasses what we have in the US. And the ability then to interrogate those databases and understand whatever the answer to whatever question you're asking is so much greater. So I mean, we just have a lot more work to do in this area, and I think what Verily is doing, what others are doing, is very important to figure out how to get the data together to make sure we have complete data sets on individuals that all of us can look into. We're actually in a public-private partnership that takes place in Europe under the European Union called the IMI, Innovative Medicines Initiative. There are real opportunities to pull this together. We're actually leading a call right now in the diabetes space, not cardiovascular space, specifically on hypoglycemia, that is going to combine all of the clinical trial databases from Lilly, Sanofi, and Novo in regard to glycemic control, to be able to then study what predictors are or what other elements are that can reduce the risk of hypoglycemia and help better define it at a population level, at least in the clinical trial database, and then expand it beyond that. So, good. That's a very important initiative. Alan, thank you for doing that. When we got instructions about when we were done, I was told there would be a flashing red light. I neglected to tell our guide, the stage manager, I'm red-green colorblind, so we may have been way over here. And for that, I apologize. There are zeros up on the time left, so I think we are done. And with that, I'd like the audience to thank the panel very much for their wonderful insights, and we'll move on. Thank you.